Welcome to Learn Physiology. Today I am going to discuss on Golgi apparatus. In one line, Golgi apparatus can be said as the delivery system of the cell. It is named after the discoverer by Camillo Golgi, the 19th century Italian physician who first called attention to them. Golgi apparatus, also called Golgi complex or Golgi body, a membrane-bound organelle of eukaryotic cells, cells with clearly defined nuclei, that is made up of a series of flattened, stacked pouches called cisternae. These cisternae are held together by matrix proteins, and the whole of the Golgi apparatus is supported by cytoplasmic microtubules. It is located in the cytoplasm next to the endoplasmic reticulum and near the cell nucleus. The number of Golgi bodies varies from cell to cell. Usually, each cell contains at least one Golgi apparatus. However, it is absent in RBC. Some of the cells may have more than one Golgi apparatus as found few in protists, to 20 or more in animal cells and several hundred in plant cells. They are especially abundant in glandular cells where it is extensively engaged in modifying, packaging, and delivery of secretory substances. The major functions of Golgi apparatus are collection, packaging, and distribution of molecules synthesized at one place in the cell and utilized at another location in the cell. It also helps in packaging of substances to be secreted from the cell. A Golgi body has a front and a back, with distinctly different membrane compositions at the opposite ends. The front, or receiving end, is called the CIS face, and is usually located near endoplasmic reticulum. Materials move to the CIS face in transport vesicles that bud off of the endoplasmic reticulum. Then these vesicles are transported to Golgi apparatus and bind with it for further modifications and packaging. The transphase is responsible for releasing the packaged materials. Proteins and lipids manufactured on the rough and smooth endoplasmic reticulum membranes are transported into the Golgi apparatus and modified as they pass through it. The most common alteration is the addition or modification of short sugar chains, forming a glycoprotein and glycolipid. Periodically, the membranes of the cisternae push together, pinching off small, membrane-bounded secretory vesicles containing the glycoprotein and glycolipid molecules. These vesicles then move to other locations in the cell, and distribute the newly synthesized molecules to their appropriate destinations. So, in brief, the function of Golgi apparatus is to process, package, label and deliver proteins and lipids. Delivery of proteins and other molecules, like lipids to different parts of the cell or are secreted by the cell membrane via exocytosis, when these secretory vesicles fuse with the cell membrane. Thank you very much for watching the video. If required, you may share this video to others and subscribe the channel if found suitable.